So I've selected two stories, which I thought might be of interest to the center here. I think one is basically the birth of proteins, and the other one, the death of proteins. I think there was a period, basically in the late 1990s, that it became fashionable at conferences to have a session entitled "The Birth and the Death of Proteins." And I was very unhappy that my part was always the death of proteins. It was typically, Paul Ziegler, who had done the structure of GROEL, who talked about the birth of proteins. And so I think today I will begin with the birth of proteins. It's an observation of chaperonin complexes in the act of protein folding in situ and in vitro. Now, I think that goes back a long, long time. I think that was more than 30 years ago in a collaboration with Ulrich Hartl, who at that time was at Sloan Kettering. We began to work on GROEL. And I think that was, well, this was still the age of blobology, obviously. So, resolution was pretty minimal. It was all negative stain, of course. But I think, nevertheless, the structure, was quite revealing. It showed basically that the co-chaperonin GROES here, at that time, there was no crystal structure of GROES, only of GROEL. Formed, a kind of, it kept basically the GROEL complex, and it enclosed basically a cavity. And I think in that cavity, what we saw was a very fuzzy sort of density here. And so it took a little bit of courage to suggest that that was the substrate, but it turned out to be right. I will show now, I think it's also an example of, how cryo-EM has really developed. So that is a more recent structure, and I will go through it in a moment in detail. We start here with the single particle analysis. We had a well-defined system at the time. We took the first data I just showed you. That was just GROEL isolated from E. coli. Now we had a system in which we had GROEL, GROES, and we had a well-defined substrate, methyltransferase, MET-K. And I think it was a very well-defined system. I think basically it was clear from mass spec that we had basically full occupancy of GROEL with the substrate. And so then we did a single particle analysis structure. That is now a structure at a resolution of something like 28 angstroms. I should say that the complex exists in two forms. That is important. Basically a symmetric form in which, both ends of GROEL are kept by GROES, but also an asymmetric form in which only one end is kept. So in blue, you see the GROEL. You see at the bottom here, you see GROES in purple. I think in a minute, you get a view of the inside where the substrate is shown in cyan. So tilt it now, and we look from inside the GROEL complex into the cavity. And in cyan, you see basically where you see, MET-K. So the question is, what folding state has MET-K achieved here? And that you see more clearly. I think I don't want to go much into detail. There's a lot of information here about the contacts of the substrate with, the GROEL complex. But you see on the bottom right, you see basically a superposition of the crystal structure of MET-K and the structure inside the GROEL complex. So basically, the only difference is here, this loop here, which is not yet in a canonical alpha helical conformation, which in the mature complex, it assumes an alpha helical, canonical alpha helical structure. Obviously or probably, it is only upon release from the GROEL that it switches in that loop into the final conformation. Or the crystal structure, of course, is from the functional form, that is the tetramer. So it could also be that during assembly into the tetramer, it reaches the final folding state. Now, we move now in situ. So this is a tomogram here of the E. coli cell. And what you see then basically in yellow, you see all the ribosomes. The ribosomes are about 30 times more abundant than GRO-EL, and GRO-EL exists, of course, in two configurations, the symmetrical and asymmetrical form here. So I think then we did a couple of experiments manipulating the conditions of growth, starting with wild type grown at in exponential phase. And then I think after heat shock, 2 hours of heat shock, still in the exponential phase, then MET-K now being overexpressed in the cell, and then finally massive overexpression of GRO-EL alone without GRO-ES. So then we analyzed the tomograms, and we wanted to know what are the most abundant forms. I mean, or what is the percentage of a single capped, asymmetric or symmetric complexes. And you see the results here. I mean, basically, it's pretty independent of the growth conditions. It's always typically something like 60% or so is in the asymmetric form. GRO-EL alone does never exist. I mean, it exists only under conditions if we massively overexpress GRO-EL without overexpressing GRO-ES. So that is, I mean, a non-physiological situation here. So that clarified a couple of things. Was highly controversial. What was, I mean, the important form, the functional form. So we wanted to address the question, can we see the substrate also in the tomograms? That was a bit of a challenge on the image processing side of things. So what you see here is basically, on the left panel here, we have the asymmetric complex. So you see basically that is a cross-section through the single particle structure here. That is a subtomogram average of an in vitro situation, and that is in the cell here. And you see we have density, very clear density here at the same position as we have in the single particle structure. So I think basically what happens is only if the substrate is properly folded and becomes compact, I mean, we have clear density, and we can say that under those conditions, basically only something like 30% or so of the cavities is filled with folded substrate. And the substrate is not yet folded. I mean, we have also density, but it's pretty diffuse there. And in the symmetric complexes here, again, the situation is similar, something like 30–40% or so has in one of the cavities, I mean, folded substrate. But there's also a subpopulation of something like 10% or so, in which we have folded substrate in both chambers. So much to the GRO-EL. Now I come to the protein degradation path. That is, I mean, the work on the proteasome. We have been working on that again also for something like 30 years or so. GRO-EL, the proteasome operates at the executive end of the ubiquitin proteasome pathway of protein degradation. It is basically a non-specific protease. Specificity comes from the ubiquitin system. I mean, proteins targeted for degradation become ubiquitinated, and then they are recognized by the proteasome. Now, the proteasome consists of two basic complexes. I think one is the core complex. We got a structure of that 30 years ago by X-ray crystallography at the time. Now, by single particle analysis, it is an effort of a day or two. At that time, I think it was 5 years of crystallization trials, something like that. Now, I should say, basically, maybe I'll go back for a moment. Protein degradation takes place here in the core particle. There's a cavity here between the beta subunits. That is where the active sites are located. So any substrate to be degraded must be able to enter the cavity here in the core complex. Now, that depends on the presence of a regulatory particle. its role is basically to recognize ubiquitinated proteins, to deubiquitinate them, then to unfold them, and also to assist in the translocation into the core complex. So basically, we have in the regulatory particle 3 subunits, which are ubiquitin receptors. Then we have a module of subunits, which consists of a deubiquitinase that removes the ubiquitin, because that is important, otherwise it cannot be unfolded. And then, there is in blue, that is a AAA ATPase that basically drives all the conformational changes the proteasome is undergoing. What you see in the following is basically the conformational changes in the functional cycle of the proteasome. Well, I think there's the ground state in which basically it is not engaged with substrate. Then there's a prime state when everything becomes irreversible. And then, there are various substrate processing states. Now, it's important for the remainder of my talk that the conformational changes are of such a magnitude that even at relatively low resolution, let's say something like 15, 18 angstroms or so, we can, decide whether a molecule is in the ground state or in the substrate processing state. So we can infer from the conformation the activity status of the proteasome. Now, next question is, where in the cell are the proteasomes? And I think this is from older work, from Sarada Albert and Ben Engel at the time, that they used, Chlamydomonas as a system. Chlamydomonas has a few advantages. It's a little bit less crowded than other cells. And on top of that, it has an almost deterministic topology of the organelles in the cell. So you can predict where in the cell, let's say, is the Golgi apparatus, where is the endoplasmic reticulum, etc. Now, in this column here, you see basically in green the positions of the proteasomes in the cell. So basically, here it is, this is the nucleus here. And so you see that the proteasomes basically line, the inner membrane of the nuclear envelope. And then we have 2 spots here. That is in the position of the endoplasmic reticulum. We'll first zoom into the situation at the endoplasmic reticulum, again, because of the deterministic topology here. Is very easy to zoom in on the Golgi and here on the endoplasmic reticulum. Now, of course, the endoplasmic reticulum is, started with ribosomes. And I think what I'm telling you in the next few minutes is basically what happens here on the endoplasmic reticulum. I think when the ribosomes, make protein, the protein is transported into the ER, where it's supposed to be properly folded with the assistance of chaperones. But the folding can go wrong, especially under stress conditions. And then I think the proteins are misfolded, but there's no powerful degradation machinery in the endoplasmic reticulum. So what happens is there is the so-called ERAT system, that is, ER-associated degradation, which means that the misfolded protein is retrotranslocated into the cytoplasm and where it becomes ubiquitinated and degraded. So that is now, well, the system has been extremely well studied in terms of the molecular biology, but little was known about the structural organization. So you see here the surface of the endoplasmic reticulum in dark blue. These are the ribosomes sitting on the translocon. And then here in this region, which is completely devoid of ribosomes. And then we have here a cluster typically comprising something like 30 or so 26S proteasomes. Now, we wanted to study that in more detail here. And so basically what we have done, that is just, showing again that there are these clusters here completely devoid of ribosomes. There's a AAA ATPase CDC48, which has been implicated into the retrotranslocation And the idea has been in the field that CDC48, I mean, does the translocation and then hands over the substrate to the proteasome for degradation. But I think our data indicates something different. So what we have done here is in terms of the analysis of these clusters here, have not used here template matching, but what we have done here is we did some sort of, we picked all the densities, we did some sorting and classification according to similarity, structural similarity. So we had subpopulations here, which we could average and then map back to the tomogram. So that is what we have done here. So we look here at a region. I mean, it's always the degradation system on the side opposite to the site where, I mean, the vesicles leave the endoplasmic reticulum. So you see here one cluster, and you see in red, I mean, the 26S proteasome, and in yellow, CDC48. Somewhat surprisingly, not directly in the vicinity of the ER membrane, but more at the periphery of this cluster here. So okay, so the question is, I mean, how good are we in picking all the molecules, and how, I mean, does, let's say, this kind of visual proteomics compare with, I mean, proteomics based on mass spec? I think the agreement is pretty good. I think it's clear that we picked basically all the ribosomes here. CDC48, even though it's a lot smaller, I mean, we have also been pretty good, but for some reason, which is not yet clear to us, I think we lost some of the proteasomes. Now, if we look a little bit more closely, we can say, okay, I mean, we can very easily decide on the assembly state. Similar to GROES, we can have symmetric and asymmetric complexes. We can infer from the conformation whether a particle is in the ground state or in the substrate processing state. We can quantify that. Now, interestingly, if you get further away from the membrane of the endoplasmic reticulum, then I think there are very, well, relatively few, maybe something like 17 or 14 or whatever percent are in the substrate processing state. But those in close vicinity within, let's say, 20, 14 nanometers, I mean, that is about the length also of the 26S complex. I think 50% or so are in the active state. Now we can take that population and do some subtomogram averaging here. And what we see is that, I mean, there's some density associated with the proteasomes, which is not accounted for by the proteasome itself. And so we tentatively, you see, I mean, that density here is quite variable. Not surprising, we think it's the substrate in the process of being extracted from the endoplasmic reticulum. And of course, there's a variety of substrates, it's not one molecular species. Now, I think much of the remainder of my talk will deal with proteasomes in the nucleus. So that is basically the raw data that is already a couple of years back when we did that work here. So we see already in the raw data without any post-processing, basically you see the typical features of the 26S complex here. And that was, well, a relatively low resolution subtomogram average here. Now, the nice thing is, again, we keep information about where all the molecules came from, and we can map them back. So initially, we show basically all in light blue, all the 26S proteasomes here. That is the nuclear pore complex. Here we are in the cytosol. There's the mitochondrion, obviously. Then we can say which one is single capped or double capped. We can say in green which one is in the ground state, which one is in the substrate processing state, and we can also say what the interactions are. Now the interactions are quite interesting in a way. This is the nuclear pore complex here. So we see we have basically two populations here. One population in red here that is in direct contact, as a matter of fact, with the membrane, but it's at the periphery of the nuclear pore complex. The yellow population is quite some distance above the membrane. That is on the level of the nuclear basket. You see that here more clearly. So we can, I mean, for individual complexes, map basically the interactions with the 26S complex. Now, I think that is all work that was published 4 or 5 years ago. Now is a story that is pretty new. I think it's not yet complete, but I think it's an interesting case, and given your interest in yeast, I think I should show it here. So, I mean, there was a question, proteasomes are great consumers of ATP. I mean, that's required, I mean, by the ATPase ring here, which drives the substrate alignment for deubiquitination. I mean, the substrate has to be brought down to the side of the deubiquitination module, RPN11, RPN8. Then I think it's required for gate opening, I mean, to get access into the core complex, for substrate unfolding and translocation. It's quite a consumption of ATP here. So the question is, what does the cell do under conditions of starvation, and there is very little ATP around? And there has been an observation by people like Kortela-Enekel in Toronto here, that is under energy-rich conditions, when cells proliferate here, then basically, as you see here, I mean, all the proteasomes are in the nucleus. I mean, you see very clearly the superposition here of the nucleus, I mean, of the proteasome signal with, I mean, the nucleus. Now, under conditions of starvation, the situation changes dramatically. Basically, I mean, all the proteasomes exit from the nucleus and they accumulate in punctae, which are referred to as proteasome storage granules here. So the key question was in the beginning, what is the organization in the proteasome storage granule? Now, I think there had been ideas around, and I think it was known that there's no ubiquitin in those proteasome storage granules. I mean, that was shown by Max-Planck. It was also shown that, I mean, in these storage granules, the proteasomes are not active. And so the idea came up that I think they were inactive because they were disassembled into the core complex and into the regulatory particle. Now, I think that seemed kind of logical because it's known that under conditions of low ATP levels, I mean, the complex falls apart. So when you isolate the proteasomes, you have to keep always the ATP level relatively high. So that was one option, and we considered also the possibility that they had not fallen apart, but there was another mechanism to keep them inactive. So we studied basically, I mean, what happens, I mean, if you go from a proliferation state to a logarithmic growth state to really quiescent upon prolonged starvation here. So in the proliferation state, I mean, the proteasomes are in the nucleus, they are dispersed here in the nucleoplasm. Most of them, as I showed before, in the vicinity of the nuclear envelope and of the nuclear core complex. Then something very surprising happens at the beginning of starvation here. Then very suddenly, I mean, the hollow complex, which is a 2.5 megadalton complex, assembles further. It assembles into trimers of the hollow complex, which in the trimer of the hollow complex, I mean, basically the proteasomes are arrested in the ground state. So it's a very convenient way of inactivating them. You will see that in more detail in a minute. And so basically what happens is they form these trimers of the hollow complex here. Now, initially, they are still kind of, the trimers are dispersed in the nucleoplasm. Then, I mean, as starvation goes on, they begin to congregate here at the nuclear envelope. Now, that is now an average of the nuclear 26S complex under those conditions. You'll see very clearly that's a reasonably high resolution map, 8 angstroms or so. I mean, the trimer formation here, arrested completely in the ground state, unable to undergo the conformational changes which are required to go through the functional cycle. So when you look take a closer look here, the trimers, I mean, assemble all here or congregate here at the nuclear envelope. Most of them in close vicinity, you see that is the clear majority of nuclear pore complexes here. Now, I think there's another phenomenon that was completely unexpected. They become tethered to the nuclear envelope by a very flexible tether. Now, the identity of the tether is still unknown. We have a couple of candidates. I mean, it must be something that can form probably a coiled coil. It has to have a membrane anchor. It should have nuclear localization signal. So I think it should be, well, we have a couple of candidates, and we are currently trying to log out of them to see, I mean, to identify the tether, which is something like, well, 27 nanometer long. So subtomogram averaging is here not very helpful at all. I mean, it's far too flexible here. But what we see when we do a subtomogram average of those that have a tether, and that's the majority of those close to the nuclear envelope, we have some extra density here, which we interpret as basically the attachment site of the tether. The attachment site here is between the second largest subunit, RPN 2 and RPN 11, which is part of the deubiquitination module. Now, what happens, I mean, if they really go on prolonged starvation into quiescence here? Now, I think initially, I mean, some of the trimers here assemble a bit further here and form, I mean, clusters of trimers here in the nucleus. But then over time, I mean, they all leave the nucleus. They all end up in this proteasome storage granules here. And that is a very interesting paracrystalline structure, which I will describe in a bit more detail. Okay, so they condense into proteasome storage granules here. That is a cytoplasmic one here. You see it's not highly ordered, but I mean, at least, I mean, there's quite a bit of short range order, but no long range order. So, I mean, if you do the image analysis, it's clear that I think these paracrystals are composed of trimers, again, arrested, of course, in the inactive ground state here. So, I mean, again, we have done, of course, subtomogram averaging here. That's a bit lower resolution, something like 9 angstrom map here of the trimer now in the paracrystalline array. Now, we can, I mean, docking now the known high-resolution structure into that. I mean, we can basically identify the subunits which are responsible, which make the contact between the trimers here. Now, a couple of subunits seem to be involved, but I think the dominant one is obviously RPN 9, which is a non-essential subunit. So there is another candidate, 7.1, which is an orphan subunit here. It's a smaller subunit here, but I think basically it's RPN 9. So what we have done then is, of course, we have truncated RPN 9, I mean, C-terminal truncation, to see what is the effect. Now, you see, that is basically the situation here. When you have the wild type, I mean, then they form these punctae, they form these proteasome storage granules. If, I mean, the interaction, I mean, is affected or impossible by the truncation of RPN 9, then they remain nuclear. And I think when you do then the subtomogram averaging here in the nucleus, you see they are unable to form trimers, and they're then, I mean, retained. Obviously, the trimer formation is required for export from the nucleus here. Okay, then I think in the proteasome storage granules, they stack well, head to tail or top to bottom, and they form long fibers here. The fibers, I mean, assemble further. Okay, that is a view of the interactions, I mean, between the two ends of the proteasomes here. Then well, they generate these long fibers, and these long fibers then assemble further into bundles. There are two types of bundles. One is a 5-membered bundle. I mean, it's still here. They always coexist in the proteasome storage granules. So you see, I mean, here we have a 5-membered bundle, and sorry, okay, and then here we have here. Okay, there's the 5-membered bundle, and then here we have the 6-membered bundle. It's kind of interesting. I mean, I think hundreds of man-years have been spent on trying to crystallize the 26S proteasome, I think, around the world without any success. But I mean, here, I mean, at least they form not well-ordered crystals, but they can crystallize. And that is the 6-membered bundle. Okay. So, I mean, they form, I mean, this paracrystalline array, which I think I don't need to describe in more detail here. Now, the interesting thing is, initially, when these puncta are formed, the proteasome storage granules are formed, it's a liquid-light state. And they solidify over time. And that, I mean, is explained by the formation of these paracrystalline arrays. So that is, I mean, experiments, I mean, that were done by flip analysis. Now, what happens now when, I mean, the cells begin to resume growth? Then I think there are, of course, ATP is around, and ATP induces, I mean, adoption of non-ground state conformations here. And that then, in a kind of a domino effect, leads basically to the disassembly, to falling apart of the whole paracrystalline array. So that is basically, so glucose replenishment here. Then I think what happens is basically that a few of these proteasomes begin to change the conformation, and I mean, the whole thing falls apart. So basically, then the proteasome storage granules are kind of a depot of fully assembled 26 proteasomes, which, I mean, upon resuming growth, I mean, they become very easy, very quickly available, and they are then retransported into the nucleus. So that is basically, I think, the summary here. So primers floating in the nucleus, they align here with the nuclear envelope. They leave the nuclear pore complex, and early PSGs are liquid-like, and then they gradually become paracrystalline. Well, that is basically the story, which is not yet complete. We are trying to identify the tether, but there are also a few other questions. I think we're also making mutants, I mean, point mutants instead of just, I mean, truncation, RPN9. But that is all ongoing. Now you see the layout of my slides has changed a bit. This is to give you an idea of what we are doing in my lab in Shanghai. That is basically, we are interested in pancreatic beta cells, and we are particularly interested in insulin secretory granules here. They have a couple of features in common with, I mean, synaptic vesicles, but they are a lot more complex. I mean, it's instead of something like 30 proteins, it's 130 proteins here. And I think one of our goals is basically to come up by integrative modeling with, I mean, a pseudo-atomic model of these granules here. Moreover, we are interested in the mechanism of membrane fusion, which is required for the release of insulin. I think the machinery, again, has, I mean, a couple of homologs involved from the synaptic vesicles, but again, it's quite a bit more complex. So we have, yeah. So I think the strategy is basically, okay, we can isolate them. There's quite a degree of heterogeneity. They are also a lot bigger than synaptic vesicles. The first step is we have done proteomics analysis, in situ cross-linking and mass spec in order to get proximity information, which should help then with the interpretation of the tomograms. Then I think we will use basically a strategy which we have used in a collaboration with Axel Brünger's lab recently, to identify, some of the membrane proteins here. What happened here, that was, work that was done by my former postdoc, Shanggu, who is now professor in Beijing. He had, from the preparation we obtained from Axel, we had obtained first, subtomogram average here of the V-ATPase. But then what Shanggu observed was always, there was one very distinct density in the proximity or in the neighborhood of the V-ATPase. And the question was, what is it? And the approach we have taken here was basically the following, that there were, from the proteomics analysis, we knew there were a couple of candidates. So of these candidates, synaptophysin, synaptoporin, synaptogyrine, etc., we generated AlphaFold models. And then I think we tried to dock them into, the density there. And you see basically that, I think synaptogyrine, here, I think it does not fit very well. I think 46.5% of the volume is not occupied, and only synaptophysin, gave a reasonably good fit. Now, of course, that had to be validated, and we validated it in this case by knocking out synaptophysin, and then indeed, the density disappeared. I think another group has done something very similar at the same time. John Rubinstein's group in Toronto, and they use mass spec data, I think, to identify also, but otherwise it's perfectly in agreement here. Okay, I think, as I said, we're interested in visualizing the process of membrane fusion here. So I think that was a bit challenging in terms of the FIP milling here. So I think we wanted to be able, to see basically, vesicle fusion between two cells here, or vesicle exit, across the plasma membrane in a system, where we had the cell exposed to buffer here. Now, okay, these are some of the tomograms here. It's relatively complex. It's very easy to recognize what is an insulin storage granule, because you see the crystalline insulin inside these vesicles here. You can distinguish them fairly easily from endocytosis here. And I think we have also managed, to obtain, well, in this scenario here, steps along, basically the fusion process. So on the left-hand side, we have basically the docking and priming of, the insulin storage granules here. Then I think here we are on the pre-fusion, in the pre-fusion state here. And in this case here on the right-hand side, the cargo, the insulin has been released. So we are trying to collect, a large number of tomograms of these scenarios, and then hopefully we can, get a real insight into the machinery of membrane fusion here. So that brings me to the end. I think some of the older work I showed in the beginning was work done by Sarada Albert in collaboration with Ben Engel. I think the work on GROW-ES was the work of Jonathan Wagner in collaboration with my colleague Ulrich Hartl. The work, I just talked about on the proteasome storage granules, that was the work of Hulu and Xiao Meng, and it was a collaboration with Cordula Enneker in Toronto and Brenda Schulman in our institute. And I thank you for your attention.
What I still don't understand is why it was even in the nucleus, because the components have to diffuse back into the nucleus, and that seems like just to store it. You have when you're starving, and then it has to go back out to start to doing the degrading.
The why questions are always difficult to answer. The nucleus is the main protein degradation compartment in the cell.
Yeah, because I just think.
I mean, I think I would say that typically in most cells, roughly 80% of all the proteasomes are in the nucleus.
Okay, well, that destroys the model. Okay, we'll further refine, let's put it that way. Ido, and then, Gerhard and Zane. Ido, in the back?
Hi. So, back to the E. coli. So, now with mini-flex microscopy, we're getting gradually closer to those resolutions. And one of the things that everyone realizes is how sensitive we are to the types of fixation we are applying. Are these all just flash frozen, or there is chemical fixation, or can you say anything
There's
about
no
any
chemical fixation. I mean, it's only cryofixation.
So,
I mean,
you know, or what can you either infer or say about differences in potential artifacts between freezing and chemical fixation, and in what is happening prior to all that?
I think in cryo, I think the only artifact that could happen is if vitrification fails. So if there's ice crystal formation, otherwise I think, but I think with an E. coli cell, vitrification is not an issue at all. Okay, so that can be done by plunge freezing. I think with other systems, of course, it can be challenging. I think very often, I think there's incomplete vitrification, but I think, and this requires then high pressure freezing, and that is in some cases, especially when the samples are really thick, not so easy to combine with targeted fit milling. Okay, so, but I think, well, what the artifacts are of chemical fixation, I mean, that is a difficult story. I mean, I think there are certainly, I mean, it generates interactions which are not physiological. I mean, I think it's a very slow process. You have to keep in mind, I mean, chemical fixation taking minutes or longer. And so I think, I mean, I think the molecular sociology of cells, I mean, is clearly affected if you have, I mean, chemical fixation.
So let me talk. You did say that at least for bacteria, the cryo can really be taken as least perturbative, or as a ground truth. Have you or others have looked to see what cryo cells look after fixation, fixed chemically, and then cryo? Would you tell what the artifacts are and how big they are?
That is difficult to answer in detail. I think, as I said, I mean, there is, I mean, quite a risk that I think you generate non-natural interactions by cross-linking. I think even if you do a cross-link of something like the 26S proteasome, it changes the structure. Okay, I mean, in the regulatory particle, I mean, very clearly, I think it looks different after chemical cross-linking. We tried that, I mean, because it's a labile complex, and we tried, of course, to stabilize it by cross-linking, but we avoided it completely in the end. So-called Grafit's method, I think,
But Wolfgang,
it really changed the.
didn't one time, or am I just imagining it, show an example where you froze something, took its tomogram, you didn't do the fib milling, I mean, it was a bacteria.
Yeah.
And then you unfroze it and it walked away.
Yeah, I think we certainly avoid.
It was spiroplasm melliferum, I think it was. Because you didn't have to do any fib milling, it was very thin, and you could unfreeze it, and then it was obviously still alive.
Yeah, I think once, I mean, you have chopped off half the cell, I think it's no.
No, okay,
And
I
it's,
said without fib milling.
yeah. No, I think, well, I think it is in a close to life state.
Yeah.
I mean, but I mean, it's
So you
no
go around
longer.
cutting apart.
I think, of course, I mean, bacteria can do the vitrification, and then you can do the thawing, and I mean, then, I mean, many of the bacteria are still alive, very clearly. But I mean, if you do the fib milling, I mean, then you should take half away of your body. I mean, it's, yeah.
We don't need proton.
Yeah, so I was wondering about CDC 48 lying at this periphery of the cluster, and you know whether these are active or are they dormant? Are they
Yeah, that is.
not the hurt one? Or
I mean, it's a controversial story. I mean, the idea was very clearly that, I mean, basically it's CDC48 that grabs, I mean, the protein and pulls it out of them. Yeah, okay, that is the dominant view in the field. It ignores a lot of evidence that came, I mean, a couple of papers from Stefan Jensch and others who had shown that the 26S proteasome is directly involved in the retrotranslocation. So I think CDC48 would not be around, I think, if it were not needed for something. So I think we call one process direct and the other one indirect ERAT. Okay, so I think that is being slowly accepted. Okay. So
Then you had a question?
Yeah, thank you as always, Wolfgang. Beautiful to see all the work. I have two short questions. First, on the proteasome, when you're looking at the substrate processing states, how much can you differentiate the different states? As in, is there much variation based on the primary structure of the protein that it's degrading, or essentially the density that you're seeing that's in that state?
I don't think we can see that. I think we can, I mean, basically we see something like, I think that's from our work, also the work from Andreas Martin in Berkeley, I think basically we can very clearly discriminate 6 steps in the cycle, okay? And I think basically from step number 2 on, substrate is involved. That is when the whole process becomes irreversible. But then I think there are a couple of minor conformational changes between stage 3 and 6. Yeah, I think that is basically the result of a large number of experiments modulating the conditions, presence of substrate, etc., etc. But the substrate itself, of course, remains invisible. You can, I mean, of course, I mean, you can block the translocation through the ATPase channel, and then, of course, I mean, you see density.
My second question is also short. When you're looking at the GRO-EL, when you have the symmetric case, and you're saying you have only a small case where there are two bound in the symmetric case. You said,
well, I mean, it's about 30 to 40% among symmetrical.
Are the contacts to the GRO-EL different in the single versus double bound case?
Which context do you mean? The context between GRO-EL, GRO-ES?
GRO-EL and the substrate.
Or are you talking about the tab, GRO-ES?
No. When you have the symmetric between the double,
And you're talking about the
No,
protein
I mean,
inside of it.
then I
protein
think they seem to be strictly equivalent.
they're strictly.
Yeah.
Oh, that's
I think we don't see any. I think it's a relatively small subpopulation. I think I showed you 10% or so. I mean, where we have substrate in both chambers. But I think as far as we can tell, I think